how we talked about water, let's talk about carbohydrates. So carbohydrates are made out of monosaccharides. And monosaccharides are single sugar units that are the base for disaccharides and polysaccharides. Monosaccharide that's going to be really key in this unit is glucose, which its formula is C6H12O6. It is a form of sugar that fuels respiration and is the base unit of many polymers. And it has a hexose shape. Galactose has the same formula as glucose, and it's not as sweet as glucose, but it is most commonly found in milk and cereals. The only difference between glucose and galactose in their structure, because they're both hexose, is the arrangement of hydroxide atoms on carbons 1 and 4 are flipped. And fructose has the same formula as galactose and glucose, and it's the sweetest naturally occurring carbohydrate and commonly found in fruits and honey. And unlike glucose and galactose, it has a pentose shape. And ribose, the final monosaccharide that we'll be talking about today, has a different formula. It is C5H10O5, and it forms the backbone of nucleic acids, DNA and RNA. So now that we've talked about monosaccharides, let's talk about disaccharides. The definition of a disaccharide is two monosaccharides linked together by a condensation reaction. So a condensation reaction is where it yields H2O as a byproduct when it removes an OH, a hydroxyl group from one molecule, and a hydrogen atom from the other molecule. Examples of disaccharides that you need to know are maltose, which is a dimer of glucose, and you'll mostly find maltose in beer, barley, molasses, and syrups. Lactose, which you're probably very familiar with, which is glucose plus galactose, and it's most commonly found in milk. And sucrose, which is fructose plus glucose, and it's most commonly what you know as like table sugar or the sugar that you put in your morning coffee. And finally, after disaccharides, there are polysaccharides, which consist of many monosaccharides linked together. And glucose molecules are what make up most polysaccharides, and they're often linked together that form very long and sometimes branched chains. And these branches either happen on carbons 1 and 4, which causes a straight chain, or carbons 1 through 6. And carbon molecules on glucose are linked in a certain way. It starts at the one just after the oxygen, and it goes around in a clockwise motion. And that's how you label the carbons from 1 through 6. So the first polysaccharide that we'll be talking about is cellulose. It is linking beta-glucose molecules together by carbons 1 and 4. And the glucose molecules are rotated 180 degrees to the previous one in cellulose, which allows it to form a straight chain because beta molecules have a specific alignment that they can't just be holding hands like this, but they have to be like upside down, like kind of like one's like this, the other one's like this. And cellulose is the basis of cell walls in plants because of their tensile strength. And humans actually cannot digest cellulose because they don't have the enzymes in their bodies to be able to break down the bonds between the beta-glucose molecules. Starch is another polysaccharide that we'll be talking about. You're probably more familiar with it. It is made out of repeating alpha-glucose molecules linked together on carbons 1 and 4. All glucose molecules in starch are oriented the same way, so all kind of like this. And there are two types of starch, amylopectin and amylose. Amylopectin is a chain of glucose molecules that is branched, which has a more globular shape for starch, and it's much larger than amylose. And amylose is an unbranched chain, so it forms a helix shape, and it's much shorter. Starch is only made by plant cells, and it is a good store for glucose and energy because it's easy to add or remove glucose units. Finally, glycogen is a repeating chain of alpha-glucose molecules, very similar to starch, but it's only more branched than starch, and it serves as a store for glucose and energy because it is easy to add or remove molecules. Glycogen is made by animals, including humans, and also some fungi. So in humans, it's mostly glycogen is stored in the liver and some muscle cells as a way to store excess glucose that you get from your diet, and then it's able to be accessed when you need extra glucose when you're not getting it through your diet. So now that we talked about carbohydrates, let's talk about lipids. Lipids are a diverse group of carbon compounds that share the property of being insoluble in water. So the functions of lipids vary greatly, but the main things are they can be used for structure. An example of that is phospholipids in the cell membrane. Hormone signaling, steroids are key in hormonal signaling. Insulation, fats in animals serve as heat insulators, and also fats in the myelin sheath of neurons serve as electrical insulators. Protection, triglycerides actually form a tissue layer around many key internal organs and provides protection against physical injury. So the fats on our body actually provides protection to like our good organs inside. And lipids are also used as storage for energy. So triglycerides can be used as long-term energy storage. So triglycerides are made up of 3 fatty acids with 1 glycerol molecule. Each fatty acid is linked to the glycerol by a condensation reaction. Triglycerides are used as energy storage in adipose, which is fat tissue. Fats are used as long-term storage because 1, the amount of energy released in cell respiration per gram of liquid is 2 times that of carbs and protein. 2, lipids only add 1/6 of the body mass that carbs do. Because if you kept all of our glucose stored in glycogen instead of fat, you'd weigh a lot more because carbs are a lot more dense than lipids. And 3, lipids also have secondary roles that carbs do not, such as insulating heat in the body and as shock absorbers for protection of vital organs. So we can think of how glucose is stored in the body as kind of like money and a bank. So cash in this scenario is glucose, and you spend it in cellular respiration, and putting your cash in your wallet is the equivalent of storing it in glycogen because it's a temporary storage that you can easily just pull it out and use it in cellular respiration when needed. And putting your money in the bank is like storing your glucose in fats because it's way harder to access your glucose when it's in triglycerides versus when it's in glycogen. And it's a lot harder to access money when it's in the bank versus when it's in your wallet. But your wallet can only store a limited amount of money. Your glycogen can only store a limited amount of glucose. So the rest of the glycogen that the rest of the glucose that your body does not use immediately for cellular respiration goes into fats. And another type of lipid that we'll be talking about is fatty acids. Fatty acids are chains of carbon atoms with hydrogen atoms bonded to them by a single covalent bond. It has a carboxyl group at one end, which a carboxyl is carbon, oxygen double bonded on one side, and a hydroxyl group bonded on the other side. And saturated fatty acids are a fatty acid with single covalent bonds between all of its carbon atoms. So it has as many hydrogen atoms as possible. And an unsaturated fatty acid is when the acid has 1 or more double carbon bonds. And that means because of the extra carbon bond, there is 1 or more less hydrogens available to bond with the carbons. And cis fatty acids are common in nature, and the hydrogen atoms are on the same side of the carbon atom, making it have a bent shape. And cis fatty acids have a low melting point, so it's liquid at room temperature. So traditionally you see that in oils. While trans fatty acids are artificially produced, and the hydrogen atoms are on the opposite sides of the carbon atoms, making it have a straight shape. So that means it has a higher melting point and it's solid at room temperature. So think things like margarine, which is artificially produced. Next up, let's talk about proteins. Proteins are made up of amino acids, and there are 22 different protein-making amino acids, but only 20 are coded for in the human genome. The other 2 you have to get from your diet. Each amino acid is structured the same, with an amine group attached to a carbon and a carboxylic acid group on the other side. But each amino acid has its own R group, which is an R group is just a placeholder for any sequence of combination of carbons and hydrogens and phosphorus or sulfur even, that it branches off from the carbon atom in the middle of the amino acid. And polypeptides are made up of amino acids. So they're chains of amino acids that are covalently bonded together via condensation reactions. So synthesis of polypeptides occurs on the ribosomes via translation. And a dipeptide is a molecule consisting of only 2 amino acids. And polypeptides can contain any number of amino acids, though chains fewer than 20 amino acids are typically called oligopeptides. For every n amino acids that are within a polypeptide chain, there is 20 to the power of n possible sequences for a polypeptide chain. The sequence for amino acids in a polypeptide is coded for by genes. It is stored in the base sequence of the genome and is transcribed and translated, which is the central dogma of biology, which is DNA, which is the basic genome, to RNA, which is kind of the messenger that takes to the ribosomes, and then into proteins, where the mRNA is translated into the amino acid sequence. Some proteins may consist of a single polypeptide or more than one polypeptide linked together. An example of just one protein made out of one polypeptide is lysosome, which is an enzyme that is used in the secretion of nasal mucus and tears that kills bacteria. Integrin is made out of 2 polypeptides linked together, which is a membrane protein that is used to make intra and extracellular structure connections. Collagen is made out of 3 polypeptides, which is a structural protein in tendons, ligaments, skin, and blood vessels. And hemoglobin is made out of 4 polypeptides, which transports proteins in red blood cells and binds to oxygen in the blood. Proteins are 3D structures, and this structure is determined by the amino acid sequence and its constituent polypeptides. So fibrous proteins are elongated, usually with a repeating structure, while globular proteins have a more intricate shape. Amino acids are always added in a particular order to form polypeptides. If they're added in a different order, then the structure and the function of the polypeptide would most likely change. An example of that is in sickle cell anemia, where the proteins within hemoglobin, when sickle cell anemia happens, the amino acid is actually different, and that changes the shape of the entire red blood cell to form that sickle shape, and that's what clogs up the blood vessels. And finally, with proteins, proteins are, because of their complex 3D formation, they are stabilized by bonds and interactions between the R groups of the amino acids. However, these bonds are very weak and they can easily be disrupted or broken, which changes the shape of the proteins, which is denaturation. A denatured protein cannot return to its original structure, so it's a permanent change in shape that can also change its function and make it useless. Heat can cause denaturation because temperature causes an increase in vibrations within the molecule that disrupts the bonds. And also extreme pHs can also cause denaturation because the charges in the R groups can change and cause ionic bonds between the amino acids and the polypeptides to break. And finally, let's talk about protein functions as a whole. While you don't need to memorize this entire list, it's good to know at least maybe 2 or 3 just in case on an exam they ask you. And the first example of protein functions is catalysis, which are, the example is enzymes. Enzymes are able to catalyze specific chemical functions inside and outside the cell. Muscle contraction, cytoskeletons, tensile strengthening, blood clotting. They're able to transport nutrients and gases in the bloodstream, cell adhesion, membrane transport, hormones and hormonal signaling, receptors, packaging of DNA, and immunity. Finally, the proteome is all the proteins produced by a cell, tissue, or organism. It's different than the genome, which is all the genes of a cell, tissue, or organism. The proteome varies depending on the cell's activities over time, while the genome is fixed. The proteome of each individual is completely unique because of the differences in activity, but also in amino acid sequences. For example, identical twins, they can have the same genome, but their proteome is most likely very different because they go through different activities and are most likely exposed to different environments and depending on what their body specifically needs in terms of proteins. So the environment does play a role in influencing the proteome because it influences what proteins need to be produced and the quantity of those proteins.